signature, which allowed us to perform an in vivo validation of the in vitro interferon transcriptional regulatory network co-inhibitory receptors. So that's what my talk will be. Now, it's been known for a number of years, work from Vijay Kutru and V. Regev, and we've had a program project grant, two program project grants looking co-inhibitory molecules, William Sharp for well over 25 years. That PD-1, TIM-3, LAG-3, and TIGIT are co-regulated and expressed as a module. So here we have, hopefully you can all see the pointer. I won't advance the slide while I'm doing this, but you can see that the expression of PD-1, TIM-3, LAG-3, and TIGIT on CD4 and CD8 cells, that they're modulated together. And that this is induced by IL-27. Here we have the induction of TIM-3, not so much PD-1, but LAG-3 and TIGIT by IL-27. You knock down IL-27 in the mouse, you lose the induction by IL-27. That's the upregulation and downregulation by the knockdown. Now, it's been known for a long time that type 1 interferon signatures are enriched in chronic viral infection in both mouse and humans, and that chronic viral infection induces T-cell exhaustion, really first identified by Rafi Ahmed in the HIV system and in LCMV infection. And that's associated with expression of co-inhibitory receptors, such as PD-1, TIM-3, LAG-3, and TIGIT, is interferon signature with the LCMV model, suggesting that there may be an association with type 1 interferons and these co-inhibitory molecules. So I wish to ask, do they induce these receptors? Again, here's what I showed you in terms of mouse and in our first experiments. And when I Googled in photograph of human, I swear this is what showed up, and I no way mean to denigrate mouse immunologists by showing this picture. But what one can see is that in CD4 cells, either with no cytokine, IL-27 or interferon beta, this marked induction of TIM-3, LAG-3, and PD-1 by interferons. So now we go into more depth to show this. Here's how the experiments were done. We took CD4, CD8 cells that was that were CD45 negative, positive, that is naive T cells, and stimulate them for different time points with CD3 plus minus IL-27 and interferon beta. And one can see the induction of, here's a control, the marked induction of LAG-3 and TIM-3 with interferon. Here's a control, and here's looking at TIM-3, PD-1. Here's a summary of data with TIM-3, LAG-3, and PD-1 individually, and the summary of TIM-3, LAG-3, and PD-1 positive cells. We do this marked induction by type 1 interferons, interferon beta, of these co-inhibitory molecules. But surprisingly, unlike in the mouse, where TIGIT is co-regulated, part of the module, these other co-inhibitory molecules, in human, we saw that TIGIT, here's TIGIT expression, in the presence of interferon is markedly decreased from 25% down to 12% from 28%. When we looked at RNA expression, we saw there in fact two modules, one with interferon with LAG-3, TIM-3, PD-1 increased with interferon beta, and the other module with TIGIT, the TNF receptor S9, other modules, CD160 being decreased by type 1 interference. So these data show that in humans, there are two modules regulated by interferon that in fact go in opposite directions. And here's the kinetics over time, the induction of TIM-3, LAG-3, PD-1 with the decrease in TIGIT. So just to take a step back, why do we have an interest in TIGIT? I mentioned this because under the leadership of Antonio Amora, we're about to embark upon a phase 1 clinical trial in patients with glioblastoma with anti-TIGIT or anti-PD-1 or a combination of the two, working with Jen Moliterno and led in my lab by Liliana Luca. So why an interest in TIGIT? This work goes back to 2012, work done by Esther Lozano in the laboratory. We've always been impressed with the biologic effects of blocking with anti-TIGIT. It's looking at TBET, the gamma interferon, GATA3, RF9, and RRC expression. And one can see that with anti-TIGIT antibody, there's a marked loss of these cytokines in culture. And if you knock down TIGIT here with an shRNA, you have marked increases in gamma interferon and decreases in IL-10. So comparing PD-1 and TIGIT in our hands and human systems, been very impressed with the effects of TIGIT. And also just comparing MS to glioblastoma, there really isn't a big difference between PD-L1 or PD-1 between MS and brain tumors, but there's a virtual absolute difference between TIGIT expression, particularly on the CD8 cells in patients with GBM, virtually absent in MS. Here's looking at TIGIT by flow in TILs versus blood, suggesting the potential importance of TIGIT in the central nervous system for glioblastoma. So first we wanted to work through, after that identification of the effect of type 1 interferons, wanted to work through the in vitro transcriptional regulatory network. So we use the same model that the Regev and Nir Yosef used in terms of setting up, identifying the Th17 regulatory network. And this was work done by Asaf in Vijay's lab. So we needed to have high resolution transcriptomic data to construct the regulatory network. For those of you who weren't engaged in terms of looking at RNA, now what we used to do is to take a T cell, stimulate, measure the RNA 4 hours later and say this is what it is. But we've learned that there are complex regulatory networks, and one needs to really do this with kinetics over time to construct a dynamic regulatory network. So to perform this network, we took naive CD4, CD8 cells, stimulate them, measure them at different time points with control versus type 1 interferon, did bulk RNA sequencing. We did 34 samples times 3 replicates with the same healthy donor. And we decided that rather than looking at human variation, which is significant, mediated by the genetics of the individuals, we would do what mouse immunologists do, which is pick one strain of mice and study it in detail. And we measured, we did RNA-seq, RT-PCR, and protein for flow. So this is a transcriptomic analysis of interferon beta at high temporal resolution. We saw differential expression of gene levels for 8 different time points with interferon stimulation. Here's a log2 expression. So we have differential expression patterns. We have an early expression pattern here and here. We have an intermediate expression pattern, a late expression pattern over here, and finally a bimodal expression pattern goes up, down, and back up. So in performing just transcriptomic analysis, we divided into transcription factors. So here's CD4 cells with different kinetics. These are different transcription factors. Again, we can see early transcription factors, immediate and late transcription factors induced. And we identified different co-inhibitory receptors and different T-cell related genes, both the CD4 and for the CD8 population. Again, in looking at the effect of interferon and what it does in terms of transcriptional networks is critical to look over time, because there's a dynamic change in these transcription factors and co-inhibitory receptors over time. So we identified the most differentially expressed transcription factors, and about 20 of them here, and these are transcription factors that were differentially regulated and decreased in both CD4 and CD8 T cells. And we, as a reality check, we asked if these were known interferon responsive genes. So here's the IFN responsive gene score over time, and then the green represents regulators for co-inhibitory receptors until the yellow HIV signatures in progressive patients, and then IL-27 regulators. So we want to examine these transcriptional factors